Welcome to the AtlantECO podcast, where we talk about the science behind the AtlantECO project, the Atlantic Ocean, and the human adventures experienced along the way. Here, we have conversations with guests from around the world who work together so that we can better understand, manage, and protect the ocean. So let's embark on the journey of AtlantECO and discover the world that lies above and beneath the surface of the beautiful Atlantic Ocean. Welcome back to the AtlantECO podcast, and today we are exploring one of the research areas and applications of the AtlantECO project in conversation with Tonje, Heggeset and Giang-Son Nguyen, joining me today from Norway. And we're going to discuss the topic of bioprospecting, what it is, what the benefits are, and how this is included in AtlantECO. But before we dive into the subject, let's get to know our guests. So Tonje and Giang-Son, hello and welcome to the podcast. It is a pleasure to speak with you today.
Hello, it's nice to be here.
Yeah, me too.
So in the podcast, we like to start by understanding a little bit more about our guests. And today we have two of you, which is great. So I'll start with you, Tonje. Can you tell us about your connection with the ocean and what started it for you? And maybe also walk us through your journey, which has led you to where you are today and doing what you do.
Sure. As you probably will understand, I'm a Norwegian, so I grew up at the coast of Norway on a tiny island. So I played around in the shores, in the waves, looking at everything happening there during my childhood. And my ancestors were fishermen, and they were really involved in the ocean. So naturally, it has been an important part of my life. Also, where I grew up, I can look straight out into the ocean. So I've seen how fierce it is and how exciting it is, dangerous, and also. So yeah, for me, the ocean is important, and it's always been a part of my life, even though it's a bit further away now that we live inside a fjord and not really in the open ocean area. And for a scientific way, I've always looked at pollution and environmental aspects and that being important and taking care of nature. So I early on wanted to go into biotech. How can we replace more polluting industry with more bio-friendly, environmental-friendly alternatives? So that's been a very strong driving factor in my life and in my career so far.
Excellent. And I guess same question exactly for you, Giang-Son.
Yeah, so I'm originally from Vietnam, and I live in the big city in Saigon. So it's not very close to ocean, I must admit. It's 2 hours away driving, but Vietnam has a very long coastal area, the East Sea. So it's also not very far from Saigon. And when I studied my PhD, I was in Greifswald in Germany. So it's a small city laying in the Baltic Sea. And actually, I found out that the area also called East Sea in German. So yeah, we visit the ocean pretty frequently. And then I moved to Tromsø and then Trondheim. So both in Norway, and yeah, no surprise that it's very, very close to the fjord and the ocean. So even though at the beginning I don't really have very strong connection with the ocean, but more and more I'm getting more involved in the research with the ocean, especially starting at SINTEF with many projects that connect to the ocean or the marine region, basically.
Thanks for sharing that. So as mentioned at the beginning of the episode, we're talking today about bioprospecting. So let's start discussing what it is, and maybe give some examples of applications of bioprospecting that people listening could link to or would have heard of.
Bioprospecting, according to the dictionary, is the search for plants, animal species from which medicinal drugs, biochemicals, and other commercially valuable materials can be obtained. Of course, for us, we work with microorganisms, so we pretty much want to include also bacteria, fungi, other microorganisms into this definition. So it's all about using what's available in nature and exploiting it for a commercial, industrial purpose. Of course, the most common examples are linked to medicine. I'm sure everyone has heard of opium, which is extracted from poppies. And not everybody might know about the willow bark, which contains a compound called salicin, which is very similar to the compound known as aspirin, so like a painkiller. So what's typically been happening in the past is that you're taking medicinal information that people pass down from generation to generation, and then you figure out how to extract it, how to get it purified and commercialized and produced in large volumes. Of course, in more modern times, in the medicinal directions, we try to look for new compounds, new antibiotics, new drugs, new anti-cancer compounds, anything that you can use to treat illnesses. But it's not just medicinal purposes. It's also industrial in general. For instance, enzymes, which are biocatalysts that you can use to convert stuff. So degrading materials found in wastewaters, for instance, in a process that you would typically call bioremediation. So removing polluting things and then cleaning up. Or you can convert crude materials into more easily convertible materials that you can then use to produce anything, basically, in a bioprocess, like biofuel is the typical example. And the list goes on. So the general concept is like a biological mining activity. What can you find? What can you exploit? What can you use?
So how is this done? If we turn to the science behind it, can you explain the process and how all this happens?
Yeah, there's two different concepts that you can do. You can use what is called functional screening, which means you look for an activity in nature. So you can, for instance, look for an antibiotic that is able to kill microorganisms, bacteria, fungi, and so on. You don't know what it is, but you can look for the function of killing these microorganisms. Or you can do what we call genome or metagenome sequencing and then look for the genes and then use bioinformatic tools to predict what these genes are encoding. So what kind of functions are found in these genes. And in that case, you don't need to know anything about the compound that is being produced, but you can find clusters that produce these compounds, or you can find enzymes that you can predict might have a function similar to something known or something completely different based on the structure of these enzymes that are being produced. So it's like two different directions that you can take.
So if I just even go one step back, is it a case of you go into nature, you pick samples of stuff, and then you bring that back to the lab? Do you know what you're looking for when you go sampling, or is it just a kind of a wild expedition and you bring everything back and hope to find something?
Typically, you have a purpose, but you don't know that you will be able to find something. So in SINTEF, we have had several projects where we are looking for new pharmaceuticals, like new antibiotics, for instance. And in that case, we know that the diversity of microorganisms that you find in certain areas is much higher than in other areas. So you would start looking for something which is novel, that is not well described, and then hoping that what you find expressed in these microorganisms is different than what has already been exploited by the pharmaceutical companies in the past. So that's sort of the way we do it for antibiotics. Doesn't mean that you succeed, but you try to increase the likelihood of success by going into the unknown. But if you look for instance, enzymes to degrade wood, like raw, then the remnants are from the paper and pulp industry, for instance, you would want to look at areas where these type of materials are normally degraded by microorganisms. So you can go into the woods, find the decaying wood areas, or you can go into the gut of bugs that eat these and then try to extract the microorganisms that you find there, and then hopefully find some enzymes that can do what you wanted to do. But it's like mining. So you go into the unknown, hoping to strike gold, in this case, biological gold. Sometimes you're lucky, sometimes you find nothing. So yeah,
yeah.
About increasing your probability of success.
Yeah, and I guess the more you do it, the more you have an inkling for where things could be or what you're looking for. in different organisms? I don't know. I'm just guessing. But
yeah, we know that antibiotics, for instance, are expressed by certain types of microorganisms. So you try to enrich for those if you're trying to grow them, for instance.
yeah.
So you select the growth conditions that favor these organisms over the other ones that usually don't express anything interesting. But that's just one way. But if you go for the genetic approach, you need to go somewhere new where there is no sequences in the databases already.
Yeah, and I'm going to ask more questions because I'm just interested. So is it, I guess bioprospecting has been done for a long, long time, although now it's modernized and we've got big tools to do it and big data sets. And you talked about bioinformatics and all that kind of thing. But how likely is it to find new things? Because I guess a lot of things have been explored and analyzed, but I'm just wondering, how often do you find something new and worthy of, exploitation?
We do find new stuff. The big problem, for instance, when you talk about pharmaceuticals, is that a lot of the new stuff is also toxic. So it's finding both the ones that are active and that are not too toxic to the cells. There's like a balance there. And, if for each compound that you find, or maybe you need to explore 100 or 1,000 different compounds for one to make it into the market. So it's like a big screening job. So you need a lot of original targets to find the one that might be worthy of commercialization process.
Yeah, so I guess you need a lot of patience, huh?
Oh, yeah. John can tell you a lot about that, I think.
Now, if I could add a bit, the problem of rediscovery, especially for the small molecules, is big because, as they said, that you will find the same thing again and again using either functional screening, digital bioprospecting, like using mining of genomes or sequencing data. So it's indeed a problem, and I think many people try to address those by looking into some really novel, for example, in case of enzymes, for example, people are now looking to novel font of the proteins, for example. Hopefully they can find some enzymes that carry out some reaction never seen before, have not seen or people have not reported it, for example. For the antibiotics, for example, there are many software, the program try to address that and try to look into the novel clusters, for example, and try to find out what is known there. I think in case of the clusters for producing antibiotics or bioactive compounds, is that there are a lot of bioactive compounds in the market or in the literature, but not many sequences of them have been known. So you usually end up in a situation that you found something novel, and then you try to express those clusters, and then at the end you found this is the compound that's reported in the literature before. Is that nobody have been able to find out the gene clusters that's responsible for those compounds. I mean, in a sense, it's also useful for the community, but the rediscovery problem is still a challenge. Yeah.
Yeah, it's good for the science, it's good for the knowledge, not so good for the business, right?
For society, because as we know, antibiotic resistance is a big issue these days, and it's increasing. So we are fighting an endless war against those pathogens that might end up killing us. So unless we increase the discovery rate, we have a problem in the long run. So something needs to be done here. So that's why, of course, we want to continue doing this. We still need someone to fund it, but yeah,
for sure. So I'm gonna take us back to the subject of the day, because I could ask so many questions still. But if we turn to AtlantECO, can you tell me about the specific type of activities that we undertake there and how it is contributing to the overall objectives that we have set? Because we're looking here at a specific set of microorganisms, right?
So SINTEF, we in charge of the activity of bioprospecting. So this is the molecular bioprospecting with a focus to discover new enzymes relevant for the stakeholders in the project. So we focus mostly on the enzymes that have high value in the diagnosis or molecular research. So enzymes that are active against DNA or RNA, for samples. So we start with the data mining. So we collect all the data that's relevant in the AtlantECO project. So that also include data that we will produce in the project, and also data from various projects that is under the umbrella of AtlantECO. So that is the data is hosted on Magnify and under the super study AtlantECO. So we gather the data set and we carry out data mining tasks. So basically we got the list of relevant class of families of enzymes that are interested for our stakeholders. And then we focus on there. After that, we do some functional screening to screen for the activity of those type of enzymes, because based on our experience, so combined approach of functional screening and data mining will increase the chance of finding something really novel. And then, of course, at the end, if we found some hit that is relevant, we will go to enzyme production and characterization, and then channel the enzymes to our partners so that they can look further where the enzymes are that they interest. But not only that, we will create the database of metabolic potentials and genetic signature. So basically, is the database of potential novel biosynthetic gene clusters that can produce new compounds or new antibiotics, and also the list of enzymes or the list of proteins that are relevant. So it's called database for blue economy. So then, in addition to the enzymes of interest for stakeholders, so we also extract the genetic biohazard markers for biological sensors.
Yeah, so a lot going on, AtlantECO. And so now we are 3 years into the project. Can you tell me, out of those activities, where are we? Have we got some results? Have you found anything of interest?
We start to organize our data, taking the data from Magnify and organize it in the way that we can doing the mining more efficiently later. So it's ongoing on. And also we try to geographically map the genetic potentials on the map, and to see where, for example, those type of enzymes, where they found in the Atlantic Ocean, for example, and whether we can derive some information about why those type of enzymes exist in those areas, for example. And also it's connect to the modeling part that we just start with other groups. So we found some hit that can be further characterized in silico for now, because usually you end up with a lot of potential hits, and you need to filter out which one is the most interesting for our stakeholders. And then after that, we will work with them to see, okay, which one we can select for further analysis. So yeah, I think that is the status for now after 3 years.
Excellent. And we have still a couple of years to go. So can we turn to what is still to come? What are we hoping to achieve by the end of August 2025? And I guess the work will carry on after that. So what's going to happen then?
Yes, I think the focus would be to shortlist the most interesting candidate for functional characterization, and to be able to provide the good candidate for our stakeholders, but also continue the work of the database so that can be used later after the project ends. So that will be contribution to the common knowledge for the scientific community. So I think that's the focus for the last 2 years.
Yeah, and of course the goal is that at least one or a few of the enzyme candidates that we propose to the stakeholders actually will be followed up in a commercialization exploitation plan. So that's a long-term goal. Of course, that something we find will be exploited, not just end up as yet another theoretical project where it stops in a database. Bioprospecting has a very applied potential. So if anywhere, this is where we should have some clear exploitation results.
And if we talk about that, what is the usual timescale between the time you find a hit and the time you find something on the market? How long does it take?
For pharmaceuticals, more than 10 years.
Jeez.
That's mostly because there are such a long list of verification and all these trials and toxicity studies and all these things you have to do before you're even allowed to apply for approval. So, you have clinical stage 1, clinical stage 2, etc. So things take time and a lot of money. For enzymes, it could be quicker, but it depends on the type of enzyme and how the market looks at the time you're trying to enter.
I guess also it depends on the application. If it's a human application, it takes a huge amount of time. If it's less living stuff, then it's faster.
It's pretty much depends on how novel it is. If it's something which is similar to existing things and just an improvement, then you need to, of course, show that what you're bringing to market can compete. But if it's novel and you have a major problem, there might be someone out there willing to try.
Fund, fast track, and well, all very interesting, and I hope that maybe we can catch up in a couple of years and see where we got to, and if there are enzymes of interest for your stakeholders. So all that is left for me to do is to thank you for taking the time to explain what bioprospecting is and how it is done, how we apply it in AtlantECO as well, and to wish you all the best with finding the one that will make it through.
Thank you.
Thank you very much. I think we will need that.
We hope that you've enjoyed today's episode and look forward to seeing you next time. You can follow the AtlantECO project on our website on www.atlanteco.eu, and you can also find us on Twitter, Instagram and Facebook. All the links and information on the project and on today's episode is in the show notes. AtlantECO is a project funded under Horizon 2020, a European Union research and innovation programme.